Great. Okay, so what we're gonna start with today, so the title of the webinar is Master Your Lab Inventory Management with SciNote ELN. I want to do a quick introduction into what we want to do with today's webinar and what we are planning to do in the next few weeks. So this is actually the first webinar in at least 3-part series. So today we're going to start and we're going to go through the overview of inventory lab management, some of the challenges, and we're also going to finish up with a use case. Then in the next webinar, number 2, probably somebody from the customer success team will join me. So we're going to go through more advanced features of SciNote and discuss how those can be set up. And the third webinar will most likely have one of our customers joining us, really showing a use case where they connect the different teams, different inventories within SciNote. So keep that in mind. We're going to be sending you emails about it. So first webinar in a 3-part series. And now let's jump to the agenda for today. So as already mentioned, first I'm going to do a quick lab inventory management overview of historical lab inventory management and what's happening today. Then we're going to go into some learnings, both from our prospects, our customers, and also some things we found both on, for example, Reddit and some of the research that has been done about inventory management. Then we're gonna, as I said already, go through the challenges, talk about solutions, and finish up with a business case. So we're gonna talk about a biobank, actually called Biobank, that used SciNote as a central inventory and ELN when they worked in different teams. Cool. Quick introduction into SciNote, so whoever doesn't know us. So first of all, we are the #1 ELN plus inventory system based on G2 reviews and have been for the 3 years in a row. We have more than 100,000 users around the world in very different countries, so 100 plus, very different sectors as well. Of course, majority pharmaceutical, biotech, and academia. But what we are very proud of is that we are also supporting FDA and USDA in their scientific pursuits. What we as a company stand for is really security first ethos. So making sure that we are looking that everybody in company knows what security means. So we are also ISO 27001 certified, SOC Type 1, Type 2, and Cyber Essential certified. And then bottom right side, you have some of our customers that are using SciNote inventory, ELN and inventory in their lab work. Okay, so to kick off the things and to do a quick icebreaker, we're gonna start off with a poll. I'm gonna start it, but the question will be, how do you currently manage your lab inventory? So we want to see and hear what's currently being used. I'm sure some of you are using SciNote. I'm not sure if that will be as one of the questions, options, but let's see, just a second. Where are my questions? Okay, I cannot find my questions. So if you can, you can just put the answers directly. Okay, here it is. Good. I think all of you should see the poll already on your screens. So just click, you have multiple options. So you can click on multiple ones. If your option is not in the poll, please put it in the chat. Very curious to hear what we have missed and what you have been using. Let me move the chat here. Okay, some LIMS out there. Spreadsheet. Okay, spreadsheets. Big majority of you are using spreadsheets. Okay, LIMS, ELN with inventory module. Good, great. Mostly manual or paper. Okay, that's interesting. Almost 20% of you are still using manual and paper systems. Okay, Google Sheets in the chat. LIMS. Okay, good. So third of you using ELN with an inventory module. Almost 20 using LIMS, majority of you, so almost 60% are using spreadsheets. And let me end the poll now so you can also see the results. So these are the current results that we had. As you can see, spreadsheets is the majority. We have quite a lot. I'm surprised by mostly manual or paper. None of you are using dedicated inventory software. And then ELN and LIMS are also quite close to each other in terms of percentages. Good. Okay, this was a nice warm-up, and let's jump into other things. Okay, Good. So a quick history of lab inventory management and how it all started. So pre-1970s, it was the manual era, paper logbooks mostly. You can still see that some of you are still using paper logbooks, index cards, handwritten labels, entirely manual, and also not a lot of standardization. I mean, index cards were standardized, but mostly not a lot of standardization was done there. What was probably the biggest issue with that, based on our research, and also this is true for almost all the phases that I'm going to talk about, that inventory knowledge lived in people's heads. So if somebody, the elder of inventories, left, you suddenly had a big hole that had to fill because you didn't know what's happening in the inventories, what's missing, what's not. Then in the 80s, I'm sure some of you have been there. So the first generation LIMS started to come up. So this was a first digital inventory, which was great, already different than before. It was centralized, standardized as well, but usually it really needed a lot of IT involvement. So it was not easy to change and customize it. And also updates were very rare, right? So it was a local system and it took a while to get it updated. And the biggest difference compared to the future things was that it was very unuser friendly to the scientist. Um, the next thing, I think this was a big step, was PC revolution and local databases. And what this means is, so personal computers, right? Computers became much more common, and also spreadsheets. So suddenly everybody was more used to spreadsheets, everybody was more used to computers, which made it a lot more close to the scientists. So it was more intuitive, but did require some training for better adoption. But what was very interesting here is that you suddenly started to connect workflows, right? Not just you didn't have separate inventories and workflows, but that started to be more connected. Then you have cloud-based and integrated systems. So that's in the 2000s, early ELNs as well. What was very important here is that suddenly, right, different teams, different locations could share inventories, and there were a lot more intuitive. You could actually, so this is much more modern software, much more connected, but it didn't have very good trainings usually. So the original ELNs, or the first in the market, had great software, but not really great support. And what changed in the 2020s, let's say, especially with COVID and AI coming in the last 2 years, so modern labs started to integrate different systems, so ELNs, LIMS systems, AI, equipment and instrument, in one place. And inventory became a big part of this lab's data ecosystem. What was the result is less manual errors, more real-time visibility, and compliance should be easier with these tools. But more important, and this is what was missing in the first and initial cloud-based systems, now change management is much more relevant. So people don't think just about, okay, we need a software that works, but they also think, okay, we actually need a software that the scientists will adopt, they will be able to learn it, and it's going to be easy to learn it as well. I'm going to do a quick pause. And again, just remind you, if you have any questions, use the Q&A session and also use the comment section, please. Okay, next slide is my favorite slide. For those of you that have joined our previous webinars, you probably seen a similar slide, and we really like doing this one. So what this is, these are quotes from our prospects, customers, and also from, you can see the source below, from Reddit, where different people express their emotions about inventories. And some of them are really hilarious. For example, this one, welcome to my world. We just hoard chemicals and the vague knowledge of what we have on hand is passed down from the elders. Or top right side. So the finish you can see. So remind yourself to take deep breaths and keep going. There's a lot of funny stuff out there, and it's obvious that lab inventory management is not simple because different people do different things using different systems, and there's a lot of things happening. Also, this one, if you've seen top left, so our PI wants me to be a mind reader and predict reagents they will need. So this is something we encounter often, and I'm sure some of you have seen this in your labs as well. Okay, good. What we're going to continue with, so this was a nice introduction into the next question, into the next poll. And the next question is, so what is your biggest inventory management challenge today? So you've seen some that we found and we heard about, but we're also interested to hear what you think is your current biggest challenge. So I'm sure there are multiple, but what was the biggest one? So just a second, let me start the QA session. Okay, I think you should see the question on your screen. So just again, click and let me know, and I'll share the poll at the end, so the results as well. Okay, let's see what are the ones. Okay, linking inventory, cool. Answers are piling in, so I need to increase actually. Okay, keeping inventories up to date, linking the data with experimental data. Yeah, I agree. That one is a very common one. Okay, tracking usage and consumption. I see this one has the most votes currently. And those are coming in for the same one. Okay, misplaced or untracked items. Yeah, that one is very common, especially in some equipment. What we saw, so just fun fact, and I hope this is going to be the future, RFIDs. So some of our customers are actually tagging their equipment with RFIDs. So if somebody moves it to another location, you always know where it is. Okay, so I'm going to end the poll and let's see the results. Okay, so as you can see, so keeping inventories up to date, that one has 15% of you saying that this is a bigger challenge. Then linking inventory to experimental data, I agree. This is very important, especially if it's two different systems. This is not easy to do. I see that nobody mentioned connecting inventory to instruments and raw data. Okay, interesting. But many of you, so 40%, almost 40%, said that tracking usage and consumption is a big problem. Yeah, I agree. This is something we hear about often because it has a lot of downstream issues as well. If you don't know what's been used, how much it's left, this is a big issue. What I was expecting, I expected that more of you will say that audit and compliance requirements are also the issue, but only 6% of you. Okay. Let me stop sharing this and move it away. And I'm gonna continue with the next slide. So what the next slide is about is research that was done by Merck on inventories and lab management. There are some data there that I found very interesting when preparing for this specific webinar. And some things are like, so the respondents to the survey that was done, that 90% of the scientists struggle to find storage location of consumables, even if they have some sort of tool in place. So this is relevant. Even if they're using something, either it's Google Sheet, either it's an ELN or a LIMS, they have issues with that. The second one is also very, very relevant, right? Instead of doing research, you spend 25% of your time on routine inventory upkeep. 52% of researchers acknowledge that unused or expired reagents are discarded due to spoilage, resulting in sunken costs, of course. The fourth one is interesting. So I didn't find a very good source of how they calculated this, but it's still a big, big percentage. So that 30% of companies' annual revenue might be paid as a fine. But the last one is also the one that's quite scary. So 15 minutes spent by the researcher to find a sample in the pharmaceutical lab. And the information on the right side, so this is European Commission's research, and it's not limited to science, but 10 billion, so 10.5 billion costs in the EU economy every year due to improper data management. So we see that this is a bigger issue than just inventory management. And the reason why, so I'm going to show the next slide. So expectations are very different to what reality actually has to offer. We are expected, or especially our customers are expected to do more, to analyze more samples, to collaborate with more teams. So everything is becoming more multidisciplinary. There's a lot more regulations every year, especially in European Union. And more people are also working remote. So there's a lot of things that you are expected to do, but the reality is very different. And we've seen this also with the first poll question we asked. So very disconnected tools, right? Spreadsheets, systems, everybody is using different spreadsheets. So data is missing, it's inconsistent. It's hard to trace where different samples were. It's hard to trace lineage. There's a lot of manual duplicated work happening. And this, I think, was the one that you all mentioned, or the majority of you. So poor visibility of stock, very relevant. There's a lot of costs involved with this. There's a lot more silos than we think, and time-consuming compliance and audit preparation. And of course, again, if somebody leaves the company and they were the only ones who knew how inventory is working, that's a big issue. So reality is very different to what scientists and labs are expected to do. What this means is that the future of inventory management has to be different. And what we think the future of inventory is, so first of all, it has to be really well connected with the experiment, so with experimental data. It has to allow for a very good team collaboration, and this means both permissions and sharing different. If you're sharing the reagents, consumables, you need to be able to track this in a collaborative way. Costs and budget, of course, right? So there are so many costs associated with expired and reagents and chemicals. It's easier to budget if you know how much of something and you can plan for it. What's very relevant we've seen in the past is that these inventories need to be very well integratable. What does this mean? So they need to be able to talk with other software. They need to be able to say, okay, if somebody updated something in a different software, the inventory, the central inventory has to be updated as well. Then it needs to be ready for audit and compliance. And one thing which we heard about lately, but nobody from the audience today mentioned this, that has to be connected to equipment and instruments more often, so to raw data. We've seen and heard this one a lot more in the last few weeks and months. Good. So I'm just going to do a quick overview of what we went through. So we started off with what and how lab inventory management software developed. So what was the beginning in the 70s, where we are today with the AI and ELNs. Then we covered some of the most common challenges that we hear about and that we found online. We covered what the research does and what we see the future of inventory should be and has to be. Now, what we're going to continue with, if you remember, I already said this in the beginning in the agenda. So we're going to go through a business case of a gliobank. So it's actually a biobank in Slovenia. So what they created is a centralized translational biobank that integrates glioblastoma patient samples, tumor models, and clinical and molecular data to support biomarker discovery. So you can already see that, first of all, biobank has many samples, has a lot of issues with compliance, and I'm going to talk more about this in a few slides. But I'm sure you can already imagine that this can be a very tricky use case to set up with inventory. And just to do a quick overview of what they do and what it was. So gliobank so it actually connects 4 major Slovenian institutions. So it connects National Institute of Biology. It connects Faculty of Medicine at University of Ljubljana, specifically Institute of Pathology. Then University Medical Center of Ljubljana and the departments of neurosurgery and neurology. And lastly, of course, Institute of Oncology in Ljubljana. And what they do, what they store is they have resected tumor tissues, both core and rim biopsies. They have blood plasma and peripheral mononuclear cells, patient-derived cell models, also glioblastoma stem cells, organoids, and at the end of the day, of course, clinical, pathological, and molecular, and imaging data. And you can see on the slide, so this is from their article, that if you were interested, you can also share it afterwards, but it's also available online. So you can see the different teams working in this. You can see the different samples. And all of these, you can see in the bottom left side, all of this was centralized in SciNote. So SciNote is what they wanted to have was a software-based platform where all of these teams can collaborate and track all of these samples. And I'm going to go, so I quickly did this in the beginning, but their challenges were very complex. So the first thing is their teams work in very different locations and different regulations. They were capturing the data with different systems. So that one is a big issue that we see, especially in bigger teams. Smaller labs are okay because at least you can go to your colleague and you can tap him on the shoulder and say, hey, where is this? But when you have a lab, I don't know, across the world, then this is much harder, right? So different systems and vocabularies. So that is a big issue as well that we noticed, especially if the naming system is not unified, you're going to have an issue. Then biospecimens, they do need strict labeling and they do need and require limb style organization. And they require very secure storage with real-time tracking and linkages to clinical outcomes, right? From the samples, you also at the end want to understand what was the clinical outcome of that specific sample. And I'm gonna move to the next slide to quickly show what the research showed, but I'm gonna explain it more at the end. But they did more research on DAB2, so DAB2 as one of the markers, as mentioned. So they notice a lot of different things. So how core and rim tumor cells are acting differently and what the research is there. And I'm going to go into these details at the end, so to explain it more in detail. Okay, good. So what I'm going to continue with. So this was just introduction into Biobank, what they do, what was their research. And now we're going to go into how they were using SciNote. So we're going to go through the more basic features they used and utilized and what was most relevant for their use case. So the first thing, right, all these clinical, pathological, molecular, and experimental data, they were stored centrally in SciNote. And the most important thing was they had very strict access control, which was role-based to ensure really secure use by neurosurgeons, oncologists, pathologists, biobank coordinators, and the research teams that were doing the research. And you can see on this screen, they set up these controls into who can see what, you can see who was assigned. And what's relevant as well, that it did support GDPR and also aligned with foundational biobanking guidelines, right? So very relevant compliance and audits. So they were working with samples derived from patients, so it was important that they follow the guidelines. I move over here. And you can see some of the inventories that they were using were very customized. So you do imagine, so this is one, for example, with cell lines, and what they really liked, especially in the beginning. So we sat down with them and we were like, okay, what do we do now? Do you have any templates? So this was the first important thing when they were setting up SciNote. They already had some templates they could use, so cell line templates, that they helped them set this up quickly. But at the end of the day, of course, they also had some custom columns and custom information that we didn't predict in our template. So it was very relevant that they were actually able to create these custom columns. And you can see what they were using. The most important one for them was stock. So I think all of you mentioned, or the majority of you mentioned, that tracking stock is very relevant. So this is something SciNote allowed them to do, together with tracking expiration dates and setting up notification system, right? It's not enough that you just track date and time if somebody doesn't get notified that they need to be either restocked or that they're expiring. So flexibility of the column types, but also having some templates ready so they can start working on this immediately. But specifically, stock management is what they really enjoyed and saw as a very big point when setting up the inventories. Next thing, so I'm not sure how many of you are aware of SciNote, and I mean, how many are you familiar with SciNote and our inventories? But every item in SciNote carries a lot of metadata, and this was relevant for them when they wanted to trace back where certain sample came from, what was as we said at the end, the output, the clinical output. So this is something they saw and created. So for example, you can see when they were doing different passages of samples, they could create children's, but more relevant is they were able to connect workflows, experimental data with the consumption. So in this case, you can see, so this specific item is assigned to this specific team and it was used in this specific task. They also set up one single source for locations so that they could always find, which was very customizable, so they could see the freezers. But also, so as I mentioned earlier, I think the most important lesson for them was, or based on also their article, that they had one centralized platform for note-taking, documentation, inventory management, SOPs, and traceability. So whenever somebody had to be onboarded, they had to just use this one software and see what's happening in here. Quick pause just to take a sip of my tea. And in the meantime, if anybody has any questions, again, a reminder, please feel free to use the chat. Okay, let's move on since there are no comments. Okay, next slide. Okay, so I mentioned already this briefly earlier, but the locations, right, is a very important part. So you want to know where your samples are, and you want to create a very detailed multi-layer storage location that mirrors your real-world physical inventory setup, right? You can see here location. You can see that this was the clinical lab, and this is freezer for gliobank, on-shelf one, box two, blood, right? So you can always find where it is, where it's located, and you could also then look at, am I running low? Do I need to do more vials? And so on. So very important, if you are working on multiple locations with multiple teams, you want to have this and you want to see where things are. Okay, so one feature of SciNote, and this is true for all the digital tools, search. So you want, and this is what they're utilizing a lot, you want to be able to search for everything you're working on. Either it's samples, either it's protocols, you want to find those results. And this is where they also saw a lot of benefits. So just being able to find data quicker than you are if you're using spreadsheets, if you're using Excel, or if you're using paper, this makes it much easier to find it. And what's maybe more relevant is that you can also search by barcoding, or just like if you have a barcode scanner, you can find the item quickly and know what was happening with that specific sample. And at the end of the day, again, it's in the single system, so in SciNote. Okay, I'm gonna go to some pictures. Okay, I just talked about barcodes. That's why I wanted to go to this slide. So every sample in SciNote can actually have a customizable label template, which of course they used and utilized. I think they were using Zebra printers, I'm not sure, with editable templates as mentioned, and you can then again filter the icons to quickly locate an item in your inventory. And you can see some of those printouts on the images. Good. Let me do a quick summary of what they did, how SciNote was used. So you can see that it was very important for them to create this inventory and sample management because sample lineage was very relevant, right? They were building a biobank. You need to understand where a certain sample is coming from. They had to have metadata about processing and cryo storage. They had to know and understand where a specific sample is located or consumable, right? Or reagent. And they had to have this connected with clinical and experimental records. Why? Because all of these samples, right, they are connected to clinical histories, they're connected to radiological and histopathological reports, they're connected to gene expression data, right? This was relevant for them. They wanted to see what the data says, and they were connected to derived cell models like organoids. And one of the results, what they did was, so they used SciNote effectively as a lightweight LIMS and ELN, and then use it as a biobanking platform. And the results were, so they went through around 300 glioma samples, and they saw that DAB2 so is one of the more independent markers that shows the mortality of patients, right? If the concentration was higher, also the survival rate was lower. Then, of course, as I said, so specific signatures in both tumor core and rim of the tumors, and they saw a lot of gene expression linked to therapy resistance, EMT, and immunomodulation. As I said, this is all available, and we are also happy to share this if you want, so just reach out to us and we're going to share the research they did. I see something in the chat or under the questions, so let me just double check. Okay, Sarah, it's something I'm going to cover this question at the end. So with the tablets, I think it's going to be relevant for other attendees as well. Good. So quick overview. As I said, so it was used by very multidisciplinary teams in different locations. It was used as a biobanking software. They were building a gliobank, and they did and used SciNote as a single inventory management software together with experimental and clinical data to come to a few very important real results. A few things that I want everyone to be aware of before I'm going to jump to other stuff. So just take home messages for today for this introductory webinar is inventory isn't just about items. So on a shelf, it's really about how people are working with it and how they organize their work, right? It's very important. It's people actually organizing the lab, not the software. Management matters a lot. So you have to assign responsibility. Usually it's not the software that is bad. Most often it's actually that there were no SOPs written how the software should be used. The team management failed because nobody either enforced the inventory usage or nobody set up the inventory in a way that would help the team. So, and keep that in mind, it should not be one person, it should be multiple people. You need to include everybody, especially if they're going to be using SciNote and using inventories, they need to understand why and who are they helping with that. I like this third one, and this is what I was already referring to now. So you should choose a solution that people will actually use. So simple, intuitive, that will save time. And the only way to do that is during the implementation, during the sales process, when talking with vendors, try to set this up. You need to set up inventories already during the sales. Don't wait until the implementation. Start doing that during the sales process and make sure the vendor goes with you through this and set this up. But the second thing for change management, include your team members. I've seen this countless times, and it's true for everything, not just inventory software. You need to include your people. They need to understand why you're doing this and what's going to be the end benefit for them, right? And you can use my slide from before. You're going to spend 1 hour a day less on just doing your operational work about inventories. I think this is going to be a good argument for everyone to say, okay, good. As long as I don't have to do that, I'm happy. And of course, focus on the real benefits, right? Flexibility adoption. I know I talk a lot about adoption, but this is, in my experience and science experience, the most important thing. Even if it's the simplest system. So I know a lot of you said it's spreadsheets. If spreadsheets today work for you and you can find the data and people are using it in the same way, that's maybe a good thing. Keep them. Maybe it's a smaller team. Maybe spreadsheets are a good way of using it. If you have issues, then look for a software that's flexible and people will adopt it. I hope this makes sense. As you can see, I'm very passionate about change management, but it's really after being in this space and onboarding people for a few years, I saw that this was the most important factor actually. Good. What I'm gonna do next is I'm gonna jump into the third poll. I want to hear your opinion. So I said in the beginning, this will be a 3-part series of webinars. Today, it was an introduction and we wanted to show a use case where inventories were used in a very complex system. But next time, we want to do a deep dive into more advanced features and more advanced use cases. So the next poll question will be, what are some of the features where you want to learn more? So if you're an existing customer, I'm sure you're going to see some that sound familiar to you. So just choose one that you want us to do a deep dive. And for prospects as well, you know what doesn't work today. So that's probably the answer that you're going to do. Okay, going and moving to poll number 3. So which lab inventory feature are you most interested in learning more about in our next webinar? So the middle webinar, the next one, we're going to be talking about specific features and showing use cases for specific features, not just a use case, but more in details for each of those. Just a second, let me start the QA. Okay, now it's up. We listed all of them. We have some ideas that we think we should cover, but I'm sure that you have some ideas as well. Okay, custom labels and barcodes, aliquot sequences. Okay, good. Ooh, suddenly auto answers popped up. Low stock alerts. Yes, very important. I agree. Expiration reminders. Okay, stock management is not as okay, which is interesting since in the beginning all of you said that this is the biggest issue, but now it's less. Okay, now it's getting traction. I'm gonna keep the poll open for a few more seconds. And update you real time what's happening. Okay, equipment management, calibration alerts. Yeah, that's an important one. I agree. Okay, low stock is still actually custom labels and barcodes are leading the way. Okay, good. For those that you choose custom labels and barcodes, can you just put in the chat? Are you interested in really like going into nitty-gritty in how you can develop the custom labels in SciNote? Is this what you're referring to? That would be just good information for us. Item relationships. Okay, I'm gonna slowly close the poll. Okay, the last question just came in. And check the chat, because I know somebody, okay. Oh, actually, we have a lot of questions here. Let's see. Okay, yeah. Label printer compatibility. Oh, a lot of questions. Okay, good. So let me share the results first, and then I'm going to dive into the questions. I'm going to try to cover as much as possible. But first, before we go into the questions, just check. So these are the results. So item relationships with 48%, the more requested one. So definitely, okay, one we're going to dive into. Low stock alerts, very connected to expiration reminders, and then item usage tracking. But the least relevant was actually the equipment management, so reservation of equipment. Okay, good. I'm gonna stop sharing this, and I'm gonna jump into the Q&A session. Let's see. Okay. I'm just gonna increase this because it's everywhere. Okay, so but something on that was on your slides was linking safety data sheets to chemicals in the inventory. Is that possible in SciNote? Yes. So the way you could do this is with a custom column file type, and then you can just upload that specific safety data sheets in that file type specific column. So then this will always be, again, whenever you open that specific item under the item card, you will see that SDS or security data sheet, and you can then reference it. Alex, can one create tasks based on specific sample supplies in inventory? I would probably need more information on this one. You can connect. So we have a very good system of connecting experimental data and task system with inventories, but it's actually done from the task system. So you're in the task and you assign those items, and you can also create them from there. So I'm sure this. My first thought is yes, but I may have misunderstood your question. Is it possible to integrate item location as a column in inventories? It's difficult to change an item location fast at the moment or change multiple item locations. So I'm sure this is coming from an existing user, I would suppose. As far as I know, no, it's not possible currently. But let me double check this, Andreas. I'll talk with the customer success and the product team and make sure, and somebody will reply to you with an answer to make sure that if this is possible or not. Okay, let's see. I'm gonna jump to Sarah's question because she had this earlier, much earlier than anywhere else. So they use SciNote on tablets in the lab. Can we use the camera on the tablet to scan a barcode and link the reagent material directly in the form or protocol? Yes. So the last time I tried this, I tried it with a phone, not with a tablet, but you can actually scan it with any barcode software on your phone, and it's going to filter out. So when you're on the task and when you're assigning the item, it's going to just filter out that one, and you can quickly assign it. So yeah, you should be able to do that. Oh, there's so many questions. Let's go. Okay. Adele, number 3, are templates available for different material types? How to link to data? Yeah, there are templates. So we have multiple templates, not just cell lines. So you should log into your SciNote, and under inventory, you're going to find them. But also keep in mind, if you already have something in Excel today, Adele, you can import this into SciNote's inventory very easily with just a few clicks. You just need to map your columns in Excel, columns in SciNote, and you're going to be having the same inventory you have there today in SciNote as well. I don't completely understand, Adele, your question about how to link the data. So what exactly you meant with this? So I'm going to skip that one. But somebody will reach out at the end to make sure that we get the right information to you. Alex, another question. Label printer compatibility. Yeah, okay. So we are integrated with Fluix printer and Zebra browser print. So there is a long list of printers supported by Zebra browser print. And we can, so there's a knowledge base article we can share with you. And also, if you reach out to your customer success manager, they will be able to provide you the exact information, which printers are the ones that are supported, and you will be able to use them. Okay, I'm gonna move to Sarah's question. How to make it easier for the scientists to record which reagents, materials, equipment is being used. So one thing is you can use the item template. So when you're creating a certain SOP under protocol library, you can have also results templates, item templates besides the protocol. So your SOP can already include the items being used. So this is one that I think would help a lot. So just that usually makes it easier. I think that's the one that I would suggest you to try out, including inspiration lots. So let me see. Okay, this is actually a full question. Let's see. How to make it easier for scientists to record reagents, including expiration lots. Okay, so expiration lots, calibrations, all of this is connected to the sample. So you're going to find this under the item card. And when you link an inventory item in executed protocol, can it automatically fill in the calibration due date to a table in the protocol? Okay, this one is interesting. So Sarah, if you understood you correctly, you can give me just a thumbs up or something. So you want to have, once you fill in the calibration due date, that a table automatically just calculates, okay, so in 3 months, we need to do the recalibration. Is that correct? Not quite. Okay, then what I would ask you, Sarah, so we don't go into too much details here, can you please reach out you can also reach out to me. So veet@sciNote.net, and also to your customer success manager, because I'm not sure who it is. So here's my email, and you can also use support@sciNote.net. Include both emails, because I may also lose emails while support is being monitored by multiple members of our team all the time. So reach out and let's dive into the specific use case. And in the meantime, we got a few more questions, and then we're going to finish off. So Musa, yes, you will receive these copies of this presentation, and you can use them in the future. And can SciNote remind us to take samples from customers in a specific date? Yeah, Andreas, you can set this up with reminders. So you set up reminders, and then you will get, so a specific person will get notified, hey, you need to get the sample from the customer. And I think we answered all of them. I hope we did. If any other questions, so we're going to slowly wrap this up for today. If any other questions pop up, please reach out to us. We're going to answer those in the follow-up emails. I may have also missed some. So we're going to go through the comments that you posted and to the questions you posted and make sure that we reply to all of you. And I see there's another question. Murden, is there a section for requesting the purchase of reagents, chemicals? For the purchasing department. Yeah, so you could set up the same thing. So SciNote has a quite nice system of notifications and tagging people. So the team collaboration part of it, let's say. So they would get an email that they were tagged on, I don't know, a specific sample. And when they log in, they could see the message that they need to reorder it. So yeah, this can be possible. Questions. So I'm gonna answer the last question from Ayur, and then we're gonna wrap up for today. I see this webinar was actually very interactive, so there was a lot of questions. Please send them to us in the follow-up emails, and we're also gonna try to reply to those that we didn't. But looks like we're on a great path with our webinars. And Mayur, so why stock management, stock quantity not deducted after adding new stock? That sounds like so two things. Either it's a bug or it's being used incorrectly. If you do have stock management, we didn't hear this often, at least I didn't. But so Mayur, what I would ask you to do, so please reach out to us to support@SciNote.net, and one of our team members of the customer success team will reach out and provide the information, and we're gonna do some research as to why this wouldn't deduct. Okay, good. So last thing before we finish off, another reminder. So you're gonna get emails anyway, but this was the first webinar of a three-part webinars. The second one, we're gonna do a deep dive into more advanced features. Looks like it's gonna be custom labels and stocks, I think, if I remember correctly. And the third one will be a webinar with one of our customers that uses SciNote and utilizes it on a daily basis in a bigger team. Cool. Thank you everyone for joining us. The recording will come up in the follow-up email. Again, let me know if you have any questions. You can reach out to me, or you can reach out to support@scinote.net. And of course, you always have access to your customer success manager. Thank you everyone. And I'm really happy for this webinar. I think I really enjoyed it, and we had a very diverse group. Thanks, everyone. Andreas, I'm going to type the answer. If help you with. Good. Andreas, somebody will reach out to you at the end in a follow-up email to make sure that, because I don't want to answer something because I'm not sure what exactly you mean. So one of my colleagues or I will reach out and explain what exactly is possible and what not. No, thank you, Andreas, for asking the questions. Questions are great. Okay, stop recording and see you next time, guys.